Morning, good afternoon, and welcome to today's Extractables and Leachables Executive Masterclass. Thank you all for taking the time to join us. I am Dr. Atul Nalawade, representing ICH Pharmaceutical Solutions, and it is a pleasure to present on this one of the most important topics in pharmaceutical industry regulatory landscape. Brief introduction of ICH Pharmaceutical Solutions. ICH is US FDA audited, ISO 17025 accredited analytical solutions provider with expertise in extractables and leachable studies, nitrosamine testing, glass delamination studies, and other regulatory analytical services. In drug development, we often exhaust years of scientific genius, clinical trials, and millions of dollars on perfecting therapeutic molecules. Yet, a silent but deadly hurdle repeatedly derails approvals at the very last mile: packaging compatibility and process material interactions. Today, we are tackling a critical and highly uncomfortable reality: why 60% of extractables and leachable studies trigger regulatory information requests or complete review suspensions from bodies like FDA, EMA, and PMDA. Throughout this session, we'll demystify these statistics, we'll analyze why standard one-size-fits-all testing models systematically fail, explore the scientific integration of chemistry and toxicology, and showcase how leveraging pre-validated in-house data libraries can reduce your compliance timelines from months to hours. Our goal is simple: to arm you with practical science-first roadmap to ensure your next filing navigate regulatory audits flawlessly. So let's begin. Slide 1. Our focus today is not merely theoretical compliance. We are bridging the gap between rigorous analytical chemistry, safety thresholds, and regulatory interpretation. Over the next few minutes, we'll break down the precise clinical checkpoints which reviewers scan for, practical study design strategies, and the operational value of ICH Pharma's in-house spectral library. I encourage you to note down your queries and send those on our email IDs or the contact details shared in the next slides. Slide 2. Let us look at the raw data. 60% of regulatory filings are hit with immediate information requests, specifically targeting their E&L dossier. Why is this? The crucial takeaway here is that these compliance failures rarely stem from poor laboratory execution or bad science. They stem from poorly justified study scoping. When you apply a boilerplate test protocol, you omit crucial clinical parameters. To a reviewer, an unjustified safety threshold is an automatic rejection. This doesn't just cost money, it triggers an average of an 8-month commercial delay. When your launch is pushed by nearly 3/4 of a year, the market share and financial impact are staggering. Slide 3. This brings us to part 1, the E&L regulatory and scientific landscape. Before we can design a bulletproof study, we must understand the evolving standards and what global reviewers are actively checking for when your folder lands on their desks. Slide 4. As you can see from this matrix, regulatory compliance is not a singular target. It is a highly segmented spectrum. For parenterals and biologics, under USP 1663 and USP 1664, the focus is on cumulative risk throughout the manufacturing. For inhalation nasal products, the safety thresholds known as analytical evaluation threshold or AET are exceptionally low because the patient exposure pathway is direct to the lungs. For medical devices, ISO 10993-18 mandates rigorous physical and chemical characterization. And most importantly, look at the bottom row. The upcoming harmonized ICH Q3E guideline is shifting the global expectation toward a continuous life cycle based material risk control model. If you are not designing your studies with this life cycle approach today, you are coding future risk into your pipeline. Slide 5. The most common error we diagnose at ICH Pharma is that we call the one-size-fits-all fallacy. Many sponsors treat E&L as the check-the-box activity. They send their elastomeric closures, syringe configurations to a lab, ask for standard extractable screening. Regulators generally reject those. Your study must dynamically adapt to your product's specific formulation, pH, active ingredients, inactive ingredients, clinical duration, of contact, then storage temperatures. Furthermore, look at the second column. Producing a list of 100 unidentified chemical peaks is not a compliance. It is a liability. Raw data without qualified toxicological interpretations, including specific PDE, that is permitted daily exposures, and TTC, that is threshold of toxicological concern evaluations, forces the reviewers to issue an immediate information request. Slide 6. Now let's pivot to part 2, the practical approach to study design. Let's walk step by step through the workflow required to eliminate this regulatory friction before your samples ever reach an instrument. Slide 7. A successful E&L program is executed in 4 linear phases. First, we perform a thorough paper-based risk assessment. We map out the materials of construction, the extraction severity of the formulation, and patient exposure limits. Second, we establish the protocol design, explicitly calculating AAT based on worst-case clinical dosing. Third, we run orthogonal analytical screening, utilizing multiple complementary analytical testing techniques to make sure no volatiles, semi-volatiles, or non-volatile organic compounds are missed. Finally, we run the toxicological assessment, calculating based on PDE limits for every chemical compound of concern. This creates a complete scientific narrative that regulatory auditors can navigate and approve with confidence. Slide 8. Let's zoom in on critical junction, a bridge between chemistry and toxicology. Standard laboratories run chromatograms, match the peaks against generic outdated commercial databases, and deliver a generic report. At ICH Pharma, we know that the true compliance lies in expert interpretation. An incorrect match or unidentified peak triggers a red flag. By linking high-resolution analytical platforms with certified toxicologists, we translate raw spectral data into concrete, clinically justified exposure profiles, closing the exact data gaps that reviewers target. 9. This brings us to part 3, in-house data generation and custom libraries. This is where strategic sponsor transition from defensive compliance posture to offensive time-saving operational strategy. Slide 10. Why do we advocate so heavily for in-house libraries? First, accelerated identification. If you have an internal database containing pre-validated chromatography fingerprints of common additives, polymers, or elastomers, what used to take weeks of manual spectral matching now takes minutes. Second, uncertainty control. Standard semi-quantification relies on generalized response factors, which forces labs to use massive safety margins, artificially inflating your reported extractables levels. Using validated internal reference sets cuts down this uncertainty. Third, historical clearances. If you see materials that are already well characterized in your library, you can clear them toxicologically in hours instead of starting fresh with months of safety tests. Slide 11. As demonstrated in this screening matrix, complete analytical coverage is not optional. Reviewers look for a balanced multi-detector approach. Elemental impurities must be screened via ICP-MS with near perfect capability. Volatiles and semi-volatiles are mapped via GCMS and headspace GCMS. Non-volatile organic compounds, which are often highly complex polymer additives, must be cleared via LCMS, complemented with in-house library. Our pre-validated library aligns across all 4 analytical domains, allowing us to rapidly deconvolute complex matrices and isolate compounds down to test part per billion levels. Slide 12. Let's look at how these theories play out in practice. In part 4, we examine real-world case studies to trace specific decision points that lead to approval setbacks or to rapid clearances. Slide 13. This first case study is a classic warning story. A biologic sponsor submitted an NDA for prefilled syringe application. They calculated their analytical evaluation threshold, AET, but failed to factor in analytical uncertainty factor. By ignoring AUF, their reporting threshold was too high, meaning several trace toxic compounds were ignored as background noise. During review, FDA caught this omission, flagged the lack of scientific justification and suspended the review. The sponsor was supposed to redo the entire analytical study under the corrected protocol, resulting in an immediate 8-month delay. A single omitted factor in their initial design cost them millions in delayed launch revenue. Slide 14. To make sure this never happens to your pipeline, we have compiled our strategic blueprint for flawless audits. First, define precise clinical contact limits. Your analytical sensitivity must always match your maximum daily clinical dose. Second, assess your entire manufacturing path, including downstream single-use systems, which can leach silicone oils or processing antioxidants into your drug substances. Third, ensure every peak is backed by formal toxicological calculations or expert QSAR profiling. Fourth, leverage pre-screened library data to rapidly eliminate system contamination and verify raw materials. Slide 15. This quote from Senior CMC Compliance Panel Advisor captures the core message of this presentation. Regulators do not flag E&L studies because of analytical signals. They flag them because of gaps in scientific justification and materials risk management. Finding an extractable is completely normal. What is not acceptable is failing to scientifically explain why that extractable does not pose a hazard to a patient. Slide 16. As we look to help you de-risk future projects, ICH Pharma has access to polymer additive database containing high-resolution chromatographic footprints of over 3,500 potential leachable compounds. We have best dedicated SVS processing risk evaluation methodology. If you are currently working on pipeline projects, these preemptive modules will be critical to your CMC strategy. Slide 17. We will now like to know about your experience, whether you are dealing with current regulatory information request, planning a study for complex parenteral, or evaluating single-use manufacturing components. Let us discuss how to build a robust science-first strategy together. For deep dive project reviews, you can reach out directly to me at atul@itrispharma.com. You may call on our numbers. You may visit our website itrispharma.com. Now, as we conclude today's session, we like to note here that E&L compliance is no longer just an analytical testing requirement. It is a critical component of patient safety and drug launch timelines. At ICH Pharma, our mission is to ensure that your innovative therapies reach the patients who need them without unnecessary regulatory delays. If you have study currently in the planning phase, or if you want us to perform complementary high-level review of your current packaging risk, please do not hesitate to contact us. Let's partner to secure a flawless path to market for your products. Thank you once again and have a productive week ahead, and we look forward to collaborating with you soon. Thank you.